Let's see. Okay, thank you very much. Let's see if those come up. Thanks for inviting me to talk today. This is a great session. Actually, I'm already seeing how much of what I'm going to talk about will touch on some things that have already been spoken about and maybe be spoken about later in the session today. So I'm going to talk to you a little bit about environmental epidemiology, tell you a little bit about what that is, how we do it, talk to you about some of the work that I'm doing in that realm, and sort of just give you a little bit of a flavor for this kind of work. I'm going to start, though, in a perhaps slightly odder place that you might not there. No, that is totally wrong. Let's try that again. There. Okay, so we're going to go back to World War II for a second and talk about war and science, right? So war is terrible and leads to all sorts of tragedy. It also happens to be a time of incredible technological innovation that happens. And World War II in particular was a moment of great scientific discovery and advancement. Lots of amazing things like technology that probably made these things possible and what we're doing here possible. As you're probably all well aware, it also leads to a lot of things that may not be so good for many different reasons. Things like the development of pesticides. And of course, one of the signature elements of World War II is nuclear weapons and nuclear bombs. And so both the good and the bad come from the scientific innovation. But unfortunately, at the time of World War II, it was still a time of relatively crude understanding of the environment and what these things that we were making and doing did or interacted with nature and the environment. And one of the places that came to the real fore and was sort of the beginning of what I would call the environmental movement, actually, modern times in the US, is the advent of nuclear weapons. So clearly it developed during World War II, massive destruction, obviously, where these things are dropped. But there was also this secondary concern about nuclear fallout, about stuff coming out, radiation from that exposure drifting far and wide and potentially having effects. And at the time, many people, the military included, basically said, look, you know, we've measured the levels in the air, not a problem, they're too low, they're not going to cause any concern for you. That was sort of the general thinking in the day of things like this. You know, okay, pesticides, we're done with it, well, put it in a barrel and stick it in the ground, we'll be okay, right? Some people started to say maybe that's actually not the full story and we need to think about what happens to this stuff. Strontium-90 is a radioactive isotope of a metal that actually is part of what gets disseminated after a nuclear fallout. And there were a few scientists that started to say maybe just low levels in the air that the military is telling us aren't a problem isn't the end of the story. And several of them started a very famous early study in the 1950s and 60s known as the St. Louis Baby Tooth Survey. And the reason they did that is that teeth are this really interesting biomarker that form, if they're baby teeth, when you're in utero and a little bit postnatally. And they incorporate things into them that are circulating when that tooth is developing. One of the things they tend to incorporate quite well are metals. Strontium-90 is a metal. And so they said, let's actually get all these baby teeth of kids and see whether kids are actually exposed to this stuff or not, right? A group then went on, they actually were tremendously successful. They wound up collecting some 300,000 teeth from children, mostly in the St. Louis area, but ultimately the entire United States and even abroad. And data from this went on to show basically that as the advent of above ground nuclear testing of hydrogen bombs really took off, strontium-90 levels in the teeth of these children went up, right? And so that this is actually a problem. This was fundamental data that led to the 1963 above ground nuclear bomb testing ban treaty that was signed by JFK in 1963. So it's a great example of sort of starting to turn this environmental science into policy and politics and have real impact. It also was really the beginning of this idea that we can't just do something and leave it and say it's done and we're done with it, right? Things happen in the environment, they move around. Ecology, the way toxins move in space and in the land is important, and we need to understand that. And one of the main scientists involved in this effort was Barry Commoner, and at the time he came up with his 4 laws of ecology, which I think are quite appropriate for what you just heard and a lot of what we're talking about today, which are that everything is connected to everything else, everything must go somewhere, nature knows best, and nothing comes from nothing. So I thought those were important ones to get out there. If you're interested in I'm going to go on to something else now, but if you're interested in the history of this and kind of how it really was at the very beginning of the sort of environmental movement and ecology movement in the world, I highly recommend the My Nuclear Life podcast 2022, last summer, August episode. There's a whole history on this. And if you're really interested in digging in further, the very next episode is about how I'm actually using 100,000 of those teeth to find those kids from the 1950s and 60s and see what their health is like now and ask the question of what was going on early life that we can detect in your teeth. How does that affect your health now? So we're doing that study as we speak, which includes elements on early life experiences and mental health late in life, including not exactly the PCEs, but something kind of close to the PCEs in early childhood. So I was excited to hear that part of the talk this morning. So this has now come to the point that we really think about things. And for environmental epidemiology, this is a critical piece, right? A foundational aspect of this, and environmental health in general, is even well before the health part, right? It is how do things move in the environment, and the idea that nature and environment and toxins, they interact, and we are involved with them. And what happens in nature is going to eventually affect us, and where toxicants go is going to eventually affect us. This is just an example of the mercury cycle, right? We think about things like, where does it come from? So is it coming from fossil fuel burning or waste incinerators, right? Does it leak out as effluent and seep into the land and the waters, or does it get spewed out through smokestacks as elemental mercury? It can then travel in air currents far and wide. It can get converted to inorganic mercury, come down with rain into the land again and into water bodies. Mercury is a heavy metal, so when it gets into water, it's going to sink to the bottom. The bacteria in the bottom of water bodies convert that mercury into methylated mercury, organic form. The fish start to eat that in a process that now we understand and was probably going on with the strontium-90 and those kids in the radiation exposure, is a process of bioaccumulation. So little bits in the bottom of the lake get eaten by little fish, the little fish get eaten by big fish, and so on, until you've got a much higher concentration in the big fish that we as humans eat, right? So, and the health of the fish is going to affect the health of the humans, et cetera. So we need to understand, this is a whole aspect of environmental health that we as environmental epidemiologists really need to have happen and understand for many reasons. One is, if I want to know what this person got exposed to, I can measure things in that person. That is one way, and there are positives and negatives about that we could get into. But other ways is I could try and see how much fish this person eats, or I could measure air levels of mercury that this person is breathing to get an understanding of how much exposure is actually happening. So it's a critical piece of environmental epidemiology. Now, back to sort of why we do this. I mean, we know that many of these toxicants or these chemicals have deleterious effects from animal work and in vitro experimentation, right? So many metals interfere with a whole slew of neuronal communication, from just axon transmission to neurotransmitter release to the reception of that signal on the secondary cell, and then second messenger systems. Lead, it looks a lot chemically like calcium, so it kind of disrupts calcium signaling, and all of what I just described at a neuronal synapse is very calcium dependent, so you can imagine it getting disrupted in many ways. Many chemicals like bisphenol A or BPA, you've probably heard of, or phthalates, are endocrine disrupting chemicals, so they just can block or inaccurately or inappropriately activate endogenous hormonal signaling systems that are critical for all sorts of processes in the body. Many pesticides, for example, like rotenone and cyperquat, are very potent mitochondrial complex 1 inhibitors, so they can destroy the energy-producing units of cells and create dysfunction and frank death of these cells, right? And these are just some, and these are sort of specific mechanisms. There's also relatively general mechanisms, like many of these things create reactive oxygen species and general inflammation that can also be deleterious, right? So this can happen at any time, in any place, and depending on where you're disrupting circuits, if it happens to be the amygdala or different areas of the brain that are involved in mental health symptoms, you potentially can have mental health issues that arise, or learning and memory issues that arise. It also depends on when they happen, right? So if they're happening in an adult, you're going to get a different type of an effect than if it happens in a pregnant person, where there's a developing fetus that's developing quite rapidly, lots of changes going on. That is a complete symphony of different finely tuned things going on. And if you start to disrupt any of them slightly in any direction, you can have very dramatic effects that are also potentially lifelong, given the state of the developing fetus. So it's a period of time that we're very interested in, and we certainly look at a lot. As an environmental epidemiologist, we're basically never looking at this kind of level, right? As close as I probably can get is maybe a blood sample to what's in somebody. But much more often, we're trying to figure out ways, much what I would call cruder ways, but for reasons like we can also go into, they still work. Cruder ways of assessing sort of, in general, if I took this room, how could I distribute you as to high or low exposure to some particular component? So that is a critical aspect of environmental epidemiology. how do we make those exposures? And an example would be air pollutants, right? Air pollution in the air. So we can use monitors across the United States that the EPA puts out to sort of check levels of different pollutants, and other universities or people have monitors all over the place. We can combine those actual measurements of what's in the air with geographic details and meteorological details, other things as well, traffic patterns, et cetera, put these all into a large model and predict basically at anywhere that our model covers what is the likely exposure level to particulate matter. Often we use particulate matter less than 2.5 microns, or PM2.5, which is known as fine particulate matter. How do we know, who's more exposed or less exposed? So we can develop maps here of the entire United States of exposure levels at any place as well as potentially any time, right? This is a close-up of New York City area where you can in fact see sort of the road network, and that is not a road network that is drawn into this diagram. That is that traffic is a major producer of a lot of these pollutants, so they will show up as if you are on that road, you are getting more exposure to the pollutant, right? So this is the kind of way that we decide, we estimate levels of exposure of different people to something like an air pollutant, and we can assign it to people at different times. And so we use things like this. We then take this kind of information on how do we know who's more or less exposed, and we combine it with, for example, very large studies of people, population studies, like the Nurses' Health Study, in this case, the Nurses' Health Study 2, to look at, these are the addresses of all the nurses in Nurses' Health Study 2, and we can look at their children, for example, and say, okay, well, which nurses have children who went on to develop autism, right? And we can say, all right, for that nurse, let's look, where was she during pregnancy? What is our estimate of exposure to different pollutants in the first trimester, second trimester, third trimester? And in this paper, what we found was a higher chance that the child has autism if the mother lived in a higher PM2.5 fine particulate matter area during the third trimester of pregnancy, but not in the first or second, nor did the coarser fraction of pollutants seem to have this association, right? So this is sort of the way that we go about assessing whether these exposures are related to things. And I know this is environmental epidemiology, but because of everything that's been talked about today before and will be talked about in the future, I want to say it's not the only thing we look at. We also look at things in this cohort like ACEs, experience of childhood abuse of the mother when she was a child. And we've found extremely strong associations between those experiences and her chance of having a child who goes on to develop autism. So psychosocial factors also tremendously important, and we sort of apply the same types of processes to looking at this. But I want to go back to the pregnancy period and say that it may be more than we think from first look, right? So when somebody is pregnant, we all think about the developing fetus that's in there and how vulnerable that developing fetus is to all sorts of perturbations. But if this is a female fetus that's developing, right, the reproductive cells that are going to go on to create the third generation are already present or developing and present in that developing fetus. So an exposure to someone who is pregnant has the chance of actually directly exposing 3 generations of people, right? So we wanted to explore this a little bit more. There's more interest. It's, as you might imagine, quite hard to do this in human populations because you need to track them over multiple generations. So there's not a whole lot of work. Where you've probably heard the most about it, whether you realize it or not, is the diethylstilbestrol or DES babies, the use of DES by mothers in the early part of the 20th century. But we explored some on DES as well. But one of the places, the Nurses' Health Study 2 actually gives us a chance to do this because of the way the cohort is set up. I'm going to show you just for illustrative examples the exposure of cigarette smoking. It's a little bit simpler. We don't model it with air pollution monitors and things like that. We just simply ask questions about whether people smoke. But as you probably know, cigarette smoking is a huge issue. Many women still smoke today. Many of them, it can cause genetic mutations. It interferes with DNA methylation and other sort of epigenetic aspects that were touched on earlier today as well. It's also an endocrine disruptor that can affect things. So we, again, turn to the Nurses' Health Study 2, which actually gives us a chance to sort of look at these questions. This is that map, again, of all the nurses. The Nurses' Health Study 2 was initiated in 1989 when over 100,000 nurses, originally in 14 states, but they've now moved across the entire US, were recruited. They were 25 to 42 at the time, born in these years here. And we have over 50,000 of them who have children, and of course, many have more than one children. So we're talking about over 120,000 children of these nurses. we also have a really, now, as I'll point out in a timeline in a second, I mean, the Nurses' Health Study is this large population cohort that is followed since 1989, and every couple of years they answer surveys, so we have all sorts of information about health and lifestyle characteristics. We have a really interesting aspect of it is that for a subset of these nurses, their mothers, right, so the grandmothers, were actually directly surveyed about their pregnancies with the nurse. So we also have direct reporting by the grandmother, about 22,000 of them with almost 50,000 grandchildren that came out of that. So just to sort of describe this in a timeline for the nurses, right, here is it starting in 1989. So again, cigarette smoking, we pretty much just asked via questionnaires. We did that of the nurses in 1999, asking them to report on their mother's smoking history when they were young and in fact even in utero. 2001, right here is when the nurses' mothers cohort study was done, and so the grandmothers were asked directly. Nurses were asked more basic questions about basically, yes, did your mother smoke during pregnancy or not? Did she smoke in the home when you were a child? When the nurses' mothers, the grandmothers, were directly questioned, there was a little extra detail like cigarettes per day. Did the grandfather smoke during pregnancy? Things of that sort. So we took advantage of this type of information to ask the very basic question of, if a grandmother smokes, is her grandchild more likely to have autism? And this is the basics, the bottom line of the results we get here. So just to orient you, right, this is the grandmother smoking as reported by the nurse, so her daughter, yes, no, or some of them didn't know. These are the total number of children of grandmothers in each of these categories and the number with autism. And what we found was about a 50% increased odds of having autism if the grandmother smoked during pregnancy. Interestingly, when we looked at whether the grandmother smoked in the home as when the nurse was a child rather than the pregnancy period, we didn't see that same association. And then down here are just a whole bunch of other things that we can control for in an attempt to isolate what we hope would be a causal effect, right? We are actually trying to go after a causal effect, and we sort of adjust for a lot of these things to sort of take out the possibility that they are accounting for what we're seeing. This is the main finding. Very quickly to show you a couple other things. When we ask the grandmothers directly, they also show this association, but it's much stronger, in fact, only present there in the heavy smokers, the 15+ cigarettes per day. And we don't see the association with the reported smoking of the grandfather. As epidemiologists, we tend to ask lots of questions like this, and I want to go into why, but some of the other things we asked were, if we actually accounted for the mother's own smoking, so we essentially control for that, do we still see this association? And we do. The results were essentially unchanged for the grandmother. And if we restricted grandchildren who were born after 1995, we see, if anything, a slightly stronger association between the grandmother's smoking and the risk of autism. And I'll tell you in a second why that's important, because one of the other things that I have a strong sideline in my work is sort of methods around how we interpret environmental or epidemiology studies in general, right? And we do that because ideally we would like to get at causality, right? We would like to know that if we could tell a grandmother to, if we could randomize a grandmother to smoking or not smoking, that if we did, that our result would say that her grandchild would be more likely to have autism, right? But this is an observational study. We're not in an experimental lab. We can't randomize that way, right? So we can't do that controlled experiment that makes animal work so sort of elegant, right? But it doesn't mean we stop there. What we do then is we try and say the only thing we can turn to at that point is to say we have to look at the patterns in the rest of the data and other associations and say, are they still consistent with the idea that this might be causal, or are they not, right? So we can't do the randomization, but we can look at other aspects of our data and say, what is it telling us? So as an example, we might be concerned that if a grandchild has autism, that's going to influence how her mother reports her own mother's smoking behavior during pregnancy, right? And this is not out of the question, because in 1999, when we asked about the smoking behaviors, many of the grandchildren would already have been diagnosed with autism. So could that have influenced the reporting of the mother? The issue is when we restrict to these children born after 1995, we're vastly reducing the number of grandchildren who would have been diagnosed already. And so if this were accounting for our finding, then it should no longer show up in this population, or at least be much weaker. And we essentially did not see any change, suggesting that that is probably not happening, right? So the next thing we do is we say, okay, well, what about mothers smoking? Maybe it's just that, if a grandmother smokes, well, the mother's more likely to smoke, and that's what actually is related to autism, and there is work on that in other settings, and that's absolutely a possibility here. But that's where the result of when we controlled for the mother smoking and don't see any change in the relation with the grandmother smoking, it kind of suggests that there's something else going on directly from the grandmother in some way, or at least some other connection there. A bigger one, and this is the kind of thing where we adjust for all those other things like socioeconomic status to say, can we rule out all these other things accounting for our findings? We try and do that, and we're trying to block pathways like this, but maybe we didn't do it, right? Maybe we didn't do it well enough. Maybe we didn't measure something correctly, or we forgot a critical variable, or we didn't have a critical variable, like, let's say, genetics, right? Very important. Maybe there are genes that are related to developing autism in the grandchild that also influence the likelihood that the grandmother smoked during pregnancy. But now we can also turn to other aspects of the results we get and say, well, when we look at the grandfather smoking, we don't see any association. But genetics that would cause this kind of a connection here would also create this kind of a connection here. So not seeing this, and that's also true for things like family socioeconomic status, if somehow all of our variables did not capture the right thing, right? Anything that gets affected by a variable like this in the same way that would affect the grandmother and the grandfather smoking in the same way cannot account for the association we seek, because we don't see it with the grandfather smoking. So we rule out these types of variables. And another one that sort of falls in the same line of this kind of thing is the grandmother smoking herself in the home with the child, with the nurse, right? So we did not see an association with that variable, but things like this that predict these two types of factors in the same way can't account for that pattern of results. So we can't just take one finding we have to look at the pattern in relation to all of the other findings we have to say what is a more likely or not outcome. So we're actually, it's hard to make an absolute call, yes, no, this is causal like in a randomized experiment, but we can tip the needle one way or the other in our thinking of how confident we might be in whether something is actually causal or not. Of course, the other place we can turn, and it was great that this was alluded to also, I think, by Ravi this morning, to sort of buttress our feelings of our confidence, our fuzziness about this might be actually causal, is to turn to experimental work in animals. And there is more work done in this, and you can imagine why it's a little bit easier to do these things, right? So there are drawbacks. It's not humans, right? It's also, in this case, we're looking at hyperactivity of mice, not autism, but we can look at different types of readouts in mice. But what's shown here is if an F0 mouse exposed during pregnancy to nicotine, so prenatal nicotine exposure, the PNE, in the F2 generation, which is effectively the grandchildren that I'm talking about, right, we see increased hyperactivity of children, of the mice that were the grandchildren of the exposed dam, right, in F0. Of course, in animal studies, you can also go further, and they even went to the third F3, and now saw the same issue here, that higher hyperactivity in the mice that were exposed 4 generations earlier during pregnancy, right? This generation now tells you we've actually got something inherited, because there is no cell in the F3 generation that was present in the F0 generation when the actual exposure occurred, right? They can also do experiments to sort of show that it's through a female line rather than a paternal line, although some things can go in one line or the other. So putting all this stuff together, we start to think, well, maybe we have something here that there's, the evidence is starting to tilt towards possibly these germline effects of reproductive cells being affected by exposures during pregnancy, and then therefore passing on at least to the 3rd generation. Now, to really know that it's something that affected a germline cell and then became inherited because of that germline cell, as I said, you have to go to another generation in the female line, as they did in the animal experiments. We're still waiting for the nurses' kids to have more kids. That's difficult to do in humans, but we hope to be able to do this. But the upshot of that panoply of results we see is that we're starting to feel like there is something going on here, right? That there may actually be effects on these germline cells. And I want to now take a moment to point out the importance of the germline cells and why we think this is particularly critical and important to know. I mean, many of you may know this, but let me just take a moment to point it out, right? If you've got a mutation or some effect on a somatic cell, like a breast cell in a woman that turns into breast cancer, right? That affects that tissue and that person, and it's a huge problem and a risk for that person. It doesn't typically affect other tissues unless it metastasizes and move on, but it certainly doesn't get inherited by the next generation, right? That is in contrast to germline cells. If you make a mutation in a germline cell, that is going to produce the next generation. So it can be inherited into the next generation and then affect all the cells in that other generation, because you're affecting the if you're affecting the DNA of the germline cell, that's what gets passed on to the next generation, right? But as was also brought up in Ravi's talk and others, and I think you're going to hear more today about epigenetics, is that it doesn't have to be a frank mutation of the germline cell. You can have epigenetic changes that will affect, as Ravi was talking about, the readout of genes on the genome in that germline cell that can be inherited. And this is one of the things he didn't touch on, but one of the scary and interesting and really in some ways cool, but can be a problem, aspects of epigenetics is that some of them anyway seem to be quite stable. So you can make changes and they can be inherited through cell division and passed on to next generations as you develop the next child here, right? Some of them are not, some of them are more labile, they change quickly, but others can in fact be inherited. So this has sort of led to a whole slew of interest of ours in looking at these germline cells and the possibility that environmental exposures are affecting maybe frank DNA mutations in those cells, but also very possibly epigenetic changes that then get passed on to the next generation. So we want to ask questions about whether toxicants affect these things. And there is plenty of evidence in different cell tissues that environmental exposures affect epigenetics. And also I'll point out here, particularly in relation to this day's panel and what's going on, is that psychosocial factors are very strong drivers of epigenetics on cells, right? So if we've got psychosocial stresses and the things we've been talking about, they absolutely can affect the epigenome. Do they target, some questions we're interested in are, do they target certain genes? Do we see them targeting genes that we know are important for autism, right? So maybe we don't need a frank mutation in an autism gene, but we need epigenetics patterning to change on those genes, and maybe the environment is doing that. The other very intriguing thing here again is if it happens here, it's hard to get at the ova, the egg cells of a woman for us to study. That's not easy. Sperm is easier, and we are actually trying to do that and look at those things. But the other thing this suggests is that the changes that were going on here, if they're inherited, may show up in all the cells of the next generation. So could we see traces of the exposure effects here in blood cells of the next generation, right? And look at things of that sort. From a sort of political, getting back to that policy side of what we do, there's a very important policy implication to all of this, and that is that neither the EPA or the FDA really consider germline cells at all when considering toxicant effects or drug effects, even drugs that are designed to be used during pregnancy, right? This seems like a glaring hole that needs to be filled in the political landscape. So with that the sort of brief overview of lots of stuff that I do, I just want to acknowledge all the people that really do all the work, including one of my former students and postdoc, Ron Rotem, who's in the audience, who's going to be talking as a flash talk today. He's right there. And funding from NIH and the Escher Fund for Autism. And if you're interested in more of what we do, you can visit our lab page, and there's also some media stuff and talks about other aspects of our work. So thank you very much. Yes.
I'm sorry if I missed this, but I was just wondering I was just wondering whether the nurses and the grandmothers who participated were evaluated for autism as well.
Yeah, no, that's a great question. Unfortunately, they were not. So we don't have, we have in a very small subset a nested case control of some of the nurses. We gave what's known as a social responsiveness scale to some of the nurses to do themselves, to do on their partner, and to do on the child. So it's a small subset that we have that information, but otherwise we don't, which is a problem. We can't sort of track it through those generations that way.